Oh my gosh, we're gonna talk about enzymes right now, which is it's like it's my favorite thing in the world. I think they're so cool. Okay, let's do it. So, um, enzymes, like we kind of talked about in class a bit already, are proteins that they do specific things. So they are. I should pull up a picture of a protein. We'll watch a video. We'll get there. Okay, they are proteins that perform some specific chemical reaction in a cell. So if you remember, proteins have four different structures. I'm going to review those real quick from our notes from Unit 1, the chemistry of life. Just so you can see them. This is why we care so much about proteins, because they're awesome. Okay, let me get to the right page. Awesome. Okay, so proteins have four different structures. They've got the primary structure, which is just your string of amino acids. And then they have the secondary structure, which is kind of motifs that will show up in the overall three-dimensional structure. The overall three-dimensional structure is tertiary, like 3D, tertiary, kind of same thing. Tertiary means three, tert means three. So the beta pleated sheets and the alpha helixes, you can kind of see those motifs in this tertiary structure. They saw local folding, with the same idea. The quaternary structure is multiple individual polypeptides that are joining together to form a bigger structure. So many proteins have a structural or other functional purpose. I can give you collagen would be an example. Collagen is in skin and hair and nails, and it provides kind of elasticity and more structural support. Muscles are made of protein, and they have a very specific function in that they have like these kind of interlocking pieces that move and contract, and that's like how muscles work. And those are all well and good. Those are nice proteins, but enzymes I think are the coolest thing. And enzymes are proteins that have a very specific shape, and then that shape allows them to be a catalyst for various reactions and create opportunities for reactions and strictly control what reactions happen and which ones don't. Excuse me. So the overall structure of the protein is of absolute importance. If that structure gets messed with in any way, shape, or form, the function of the protein of the enzyme will either be drastically hindered or completely ruined. So the biggest thing to remember about these is, I guess, that they have an active site. And this is a small part of the molecule. It's usually like a little pocket where the substrate, which is the reaction, the molecules that are going to be involved in this reaction will perfectly slot into. You can think of it like a highly, highly specific puzzle piece. Okay, so the substrate is the actual thing that's affected, and they're built into the active site. And like I mentioned earlier, these active sites are extremely specific. It will be a very specific substrate based on its shape. We'll get into a whole bunch of enzymes that are even more specific, like with DNA. Not only is it a specific kind of like protein or molecule that has to be, or piece of DNA that has to be in this site. It could have to do with the whole DNA code. Like these things get incredibly specific, while some are a lot more generic, just depending on their function. Excuse me. So the active site itself, that specificity is determined by the R groups. So I want to go back to our amino acids. So if you remember, amino acids have this structure. They always have 2 carbons, and a nitrogen is the backbone. Actually, it's hydroxyl group as well. So if you see a repeating strand, you would see nitrogen, carbon, carbon, nitrogen, carbon, carbon, nitrogen, carbon, carbon, repeating and repeating and repeating. Everything about this is identical except for the side chain. And the side chain is what determines everything. That determines how this protein will be able to fold, and it will determine what function it has, which areas are hydrophobic, which areas are hydrophilic, and that all determines its shape. Determining the shape is all based off of what position will put this molecule where it will have the least amount of potential energy. So twisting it into a position that's not ideal means adding like potential energy to it. Releasing it, it's kind of like stretching a rubber band. It wants to be in the most energetically favorable position. Speaking of that, this is like a whole area of like molecular biology and biochemistry research is in modeling what proteins look like and modeling what enzymes will how they actually fold. The folding is so absolutely crucial that that's what people will spend, decades studying. There's tons of people who are researching all of this. There are modeling programs where the big goal is to be able to feed in a polypeptide sequence. So like, hey, here's the sequence of this protein. And for this computer models that can accurately represent how it actually folds given different situations. Most modeling programs now are pretty good, but they don't always get it exactly right because it's really difficult and it's extremely complicated. And understanding that would help us really understand how like biology actually works and how all these cool things. I don't know. It's so cool. Anyway, sorry, I'll get way into that. But yeah, so here are all the side chains and they all have different properties. And excuse me, that helps determine the structure of the whole enzyme, but specifically the active site. So I linked a video for you, and I'll probably link it again, that goes through a really detailed example of exactly how enzymes work and how they are catalysts. That will go into way more, that video goes into way more detail than you all need to know. Actually, I'll show it to you right now, just because we may as well, right? You should absolutely watch this video. I thought it was like insanely fascinating. Let me see if I can find it here. Here we go. I was doing some research on something else. We can talk about that later. It's pretty cool. Nope, stop. Oh, sometimes Zoom, let me tell you. Okay, this video here, it's from the RCSB protein data bank. We made this. It's got some really nice models. I can kind of start playing it for you. I don't think you'll be able to hear it, and that's okay. So these are all enzymes. So enzymes are all blobs. And what you can see is that aside from the enzymes, there's just a whole bunch of other molecules floating around. So some enzymes are kind of free floating. Some are embedded in membrane proteins of various organelles or the cell membrane or who knows. And yeah, it's a pretty good picture. So the way that this actually works is that all of the cytosol, cytosol versus cytoplasm. I promised I would tell you the difference between the two, and I did find it out. The cytosol is referring to the actual fluid that's inside of a cell. The cytoplasm is more like a physical location. So you would say that like a mitochondria is in the cytoplasm, but the cytosol contains free-floating ribosomes. Or the cytosol has a whole bunch of ions and other molecules dissolved in it. It's a tiny, I can barely tell the difference, but there you go. Okay, where was I? Oh yeah, we're back to this. So the way that somebody asked me today, how does the enzyme know which substrate should go into it? And how does that actually happen? Well, the way it happens is that all of these things are just kind of free floating, and an enzyme has this active site, and any molecule can attempt to get into that active site. And they might kind of even move into it and out, like flowing just like water, and just like little particles dissolve in water, which is exactly what happens. But until the correct molecule enters the active site, nothing will happen. The enzyme won't react. The enzyme's reaction won't continue. Oh, the bell. I'm at school. Yeah. So let me just see. Yeah. So you can kind of see the active site. So the active site is the place where those little molecules went inside, and it catalyzed a reaction. So let me go back to my notes, and we can always come back to this. I'm talking to myself mostly. Excuse me. Okay, so so the catalysis over here is going back to that whole energy thing we were talking about last video. That the activation energy is how much energy it takes to make a reaction happen. It usually has to do with the temperature. So reactions happen because molecules have arrange themselves in the correct way and hit each other with enough force in the correct position to happen. And enzymes physically grab molecules and put them in the correct configuration to make that process more likely and make it it means that you need less energy of motion and just kind of randomness in order to make this reaction happen. Biological enzymes are the best catalysts that exist. They're super specific, and they do the job really, really, really well. Because that's the reason they exist, because biological organisms have evolved to get the best version of them that we possibly can, which is pretty cool. So enzymes keep working over and over again. They are not consumed as part of the reaction. They just make the reaction happen. Let's go back here for just a second. There's a couple of the words you're going to need to know. Cofactor and coenzyme, these two things are somewhat interchangeable. They kind of mean the same thing. I'll be honest with you. I took a very specific class on cofactors versus coenzyme. It wasn't versus, but it was about them. And even at the end of that, I wasn't 100% sure of the minute differences. Basically, they are parts of the non-proteins that are involved in the enzyme, that are built in as part of the structure, that are necessary for its function. Vitamins are a really good example of this. Vitamins like vitamin B or vitamin C or whatever, they're important to us because they are coenzymes that are part of an enzyme that help it do its function. Cofactors are also the same kind of definition, but they tend to be more like metal ions. So hemoglobin is in your blood, and without hemoglobin, your blood couldn't grab oxygen the way that it needs to, because there's iron in it. There's an iron ion. If iron ions were just free floating in our body, they would wreak all kinds of havoc. It would be a mess. They would just start having all sorts of stealing electrons and giving electrons to other things, and it would just cause all kinds of mad disasters. But in hemoglobin, they're wrapped up in a biological cage molecule kind of thing, which I think I would call a coenzyme. I think the iron is a cofactor, and the thing that kind of holds it is the coenzyme. I think that's kind of what I would go with. I think that's what vitamins are, but I don't remember. Anyway, you don't need to know that at all. I'm just rambling. They help make enzymes work in doing cool stuff. So you need iron, you need magnesium, you need all sorts of metals in your body in small amounts, even copper, because they help enzymes function. That's their whole purpose. Okay, allosteric sites is another thing you're going to need to know about. Allo means other, and steric means place or something, apparently. It's these Greek root words, but it means it's other, not active sites, but other places, other binding sites on the enzyme that are not the catalytic center. It's not where the actual reaction is happening, but they affect how the enzyme is used. We're going to talk more about this in the coming units. Thank God. We're going to talk more about this as we continue, but regulation is really, really important in cells and living things. So determining if an enzyme should be doing its thing or not, like turning it on or turning it off, is really, really important. And that's what allosteric sites are all about. They determine if an enzyme is going to function or not function. Sometimes you need something bound to an allosteric site in order to make the enzyme even work. Sometimes binding something to it will stop it from working. There's lots of ways to do that. It all has to do with changing the shape of the enzyme, or somehow blocking, either changing the shape of the enzyme so that therefore the active site changes and is no longer able to bind with its substrate, or by physically blocking, sorry, got your lights back on, or physically blocking the active site. We're going to come to that in just a second. So back down here. All right. Enzymes function in their correct environment. So let's talk for a second about thermophiles. I don't know if you've ever heard of a thermophile. These are bacteria or other organisms, but usually bacteria, that can live in extremely high temperature areas. There are bacteria that live in geysers. We're talking practically boiling point. There are bacteria that live in thermal vents in the bottom of the ocean. They are adapted to exist at very, very, very high temperatures. So if you take a bacteria from a very hot, that's adapted to live at a very, very hot temperature, and you take it and put it in a colder water or colder scenario, it's going to die. And one of the reasons for that is that all of the enzymes in that organism have adapted to function and have a particular configuration at that temperature. If the temperature changes, the shape of enzymes will also change. And then therefore those enzymes will no longer function, and that cell will not be able to survive without being able to do all the important chemical reactions that need to happen for it to live. The same thing is true with a lot of other factors, like pH is a huge one. So if you add way more hydrogen ions, if you lower the pH, or if you increase the pH, if you remove a whole bunch of hydrogen ions, that will affect the R groups of the protein, and it could be denatured. A lot of these other ones, like ion concentrations and substrate and product concentrations, are more about, probably more about regulation. Usually if substrate concentrations, like the thing that goes into the, I didn't even talk about this picture. This is the substrate, the thing that's being changed. If that concentration increases, the enzyme will work more quickly because there's plenty of that. Like for example, this is sucrose. If there's tons of sucrose around, the enzyme will constantly have more sucrose entering that little active site. If that concentration decreases, then the enzyme won't work as quickly because there's nothing to convert. It kind of just makes sense, right? You're at a grocery store, you got checkout lines. If there's nobody there, no one's getting, the store's not, checking out any baskets of food or whatever, getting customers through. If you've got lots and lots of customers, then everyone's working at high efficiency. Same thing with product. A lot of times enzymes have a feedback loop where if there's a lot of product concentration, a negative feedback loop where the enzyme doesn't need to work anymore because there's already enough product that already exists. It's like, hey, you've done your job, stop working. Sometimes that product will having too much product will inhibit the enzyme. Sometimes they have like an allosteric site that's connected with the product. So the enzymes will start to shut off and stop functioning because there's plenty of product. So other ion concentrations, so too much salt could definitely, sodium or chlorine atoms can definitely mess with enzymes. So the key basically is an enzyme is adapted, organisms have adapted to have enzymes that work for the situation in which they live. They're kind of homeostasis. They're ideal temperature, ideal pH, ideal everything. As soon as you start pulling organisms out of that, their enzymes are not going to work as well, and that's going to be very bad. Okay, denaturing. Denaturing means falling apart, losing their structure. So denaturing will never mess with the primary structure, because the primary structure is just the sequence of peptides. There are ways that that can get ruined, but that's not denaturing. That's like actually cutting apart that structure. This is just them falling apart. So if you imagine like a piece of string that's all twisted up, or like, you know, something that's all twisted up, and you unknot that. Primary structure is just the straight line. The big complicated knot is the secondary, tertiary, and possibly quaternary structures. The biggest things that change that are temperature change and pH change. That can really denature proteins. When you cook eggs, you are denaturing the proteins in the eggs, and that's why they go from being sort of clear and goopy to being sort of white and solid. Protein denaturation, crazy, super awesome. Sometimes some enzymes, if they are denatured and you, like, let's say you heat it up and then try to go back to a cooler scenario, they will refold perfectly, no problem. Sometimes they don't. That is often the case, especially for eukaryotic cells, because sometimes there are biological, like when a protein is being created, there are other enzymes or other molecules that force the enzyme to fold correctly. That's one of the things that like can happen in like the rough ER, is that as this protein is being, we'll talk more about that when we get to the central dogma for translation. When the protein is being translated from DNA, or sorry, RNA to a polypeptide sequence, there can be complexes that force the proteins to fold into the correct positions, because it's always possible there's more than one way that a protein could fold together. Okay. I want to go back to that video for a second just to see if there's anything cool there. I think there was a couple of things I wanted to talk about. And then we'll talk about inhibition. Okay, so right away, looking at this model here, you can see that this protein is made up of two different subunits. We call them subunits because they're subunits. So it probably is made up of two different enzymes that have come together. So that is the quaternary structure. So the whole thing would represent quaternary. So there's probably subunit A and subunit B. Subunit A has a primary, secondary, and tertiary structure. Subunit D has primary, B has primary, or subunit 2 or whatever, is primary, secondary, and tertiary, and then the quaternary is those two together. Let me just run through this real quick and we'll see if there's anything interesting. Yeah, so you can see everything's kind of floating around. We'll talk more about that. Let's see. Yeah, so this is a good illustration as well. Reactions without catalysts just go very slowly, very slowly. So here, that's how many. It's a very tiny number, like a tiny decimal point. With an enzyme, reactions can happen a lot faster, which is good. Sometimes we need things in ourselves. Yeah, this video is just freaking awesome. Cool. I'll let you guys watch that on your own. I don't think there's anything too exciting I need to share about it, but definitely do, because it's so good. It goes in way more detail. It goes in the kind of detail that I learned when I was in like high-level biology and biochemistry classes in college. So if you like that and you think that's super cool, you'll know what you have to do in the future. Okay, let's talk about inhibitors, and that'll be the last thing we talk about for now. Inhibitors are molecules that either bind to the active site or allosteric sites, which make the enzyme stop working. So here's an example, really cool. There's also activation. So an allosteric inhibition means that an allosteric site, something will bind to an allosteric site, which will alter the shape of the active site. Over here, it's the opposite, where an activator has to be added before the enzyme will function properly. I like these pictures a lot. I think they're pretty self-explanatory, but we can still go through them. So this is a good example of just a normal reaction. So this particular substrate enters, it's got that perfect shape, a reaction occurs, there you go. An inhibition is something that can block that site and it's a competitive substrate, a competitive inhibition. So that's one type. You don't really have to know a lot of these details, but it's just kind of fun to see the options. This one is cool. So this is a type, there's a different allosteric site here. This one physically blocks the active site. So this particular kind doesn't alter the shape of the active site. It just physically covers it so that a reaction can't occur. This one is another example. This one is an example of changing the shape. So it's very similar to up here. So this particular site, something can come in. The number of examples of this, of different kinds of inhibitions, like they're numerous, they're like countless. Sometimes there's a lot of allosteric sites. Sometimes there's one allosteric or one or two allosteric or a few, and many different things could bind into all of those different places. The possibilities are insane and numerous and really freaking cool. I'm gonna make a video pretty soon about some of the research I did when I was in college, because it kind of relates to what you guys will be able to understand it, I think. I was researching a bacteria that breaks down cyanide, which is carbon and nitrogen triple bonded together, that breaks it down as a secondary nitrogen source. And part of why cyanide is so bad is that it inhibits the cellular respiration by messing with one of the enzymes in the citric acid cycle, or the electron transport chain, which we'll talk more about later, and I'll go into more detail then, but it'll be pretty cool. You can see all the cool stuff that research people are doing and how all of this all fits together. General message is this stuff is insanely complicated, and you're never going to learn absolutely everything, but as long as you got kind of a general gist of what's possible and these kind of common themes or motifs, it should all kind of hopefully make sense. So enzymes, insanely important. They literally they pretty much do everything. They are the workhorses that actually make things happen in your cells, in cells in general. And not only cells, viruses, which don't have cells. They're kind of something else, but they're cool too. And structure equals function. Anything that messes with structure messes with function. I'm going to stop there because if I don't stop now, I'm never going to stop. And that's not what you want right now. That's not what I want right now. So hopefully that helps. Oh, videos. I want to go through other videos with you real quick, and then I'll stop for realsies. So obviously that video was freaking awesome. I love that one. Let's go back. Okay, great. So that How Enzymes Work is an excellent video to look at. If you're curious about activation energy, then this video might be very helpful. And I think those might be all the ones I have in that playlist. Let me just check. There's a lot of playlists here, so I get it's very confusing. Where is AP Biology? Let's look at the whole playlist. Excuse me. Yes, click. Functioning? What's happening here? Why are we not going to the full playlist? There we are. So this video, as we go down looking at this one's amazing. This is Mr. Anderson once again kind of going through more like a small lecture on everything I covered. So if you didn't quite understand what I was saying or I was super rambly, his is a lot more polished. Activation energy is quite useful. It's only 5 minutes. And then there's also at the top the official AP Biology College Board lecture from last year. You can also check that out. It's a bit long, but it's very thorough and quite nice, and even goes over an investigation, like a lab, so that's pretty cool. We're going to be doing a lab all about enzymes, which I will talk to you about very, very soon. After this, we're going to go into photosynthesis and respiration, so that'll be the next thing to do. All right, I'll catch you later. Catch you next time.